Hey everyone, and welcome to today's Explainer. Look, if you're a focused learner, and especially if you're gearing up for the CSIR-NET Life Sciences exam, mastering cellular organization isn't just a nice to have, right? It's absolutely critical to your success. So today, we're unpacking exactly how these molecular machines work, breaking them all down visually so you can lock this knowledge in for good. So think about this for a second. How does a cell actually communicate with the outside world without breaking its borders? I mean, the cell membrane is essentially an impenetrable barrier. It's a wall. But a cell isn't just some closed-off fortress, you know? It constantly needs to take in nutrients, pump out waste, and receive signals just to survive. And to do all of that, it needs some very special, highly dynamic gateways. Okay, let's dive into this. Here is our roadmap for today. First, the cell's dynamic border. Then, anatomy of a transmembrane protein. Next, proteins in action. And finally, CSIR-NET quick facts. All right, Section 1, the cell's dynamic border. Now, before we can really understand the proteins themselves, we have to understand their neighborhood, which is the phospholipid bilayer. Picture this not as a rigid, static wall of bricks, but more like a fluid mosaic, a constantly moving, shifting layer floating in an aqueous environment. This bilayer is made up of two totally distinct parts. You've got the hydrophilic heads, which absolutely love water, and the hydrophobic tails, which repel water and face inward, hiding away from the moisture. And because of this setup, we essentially have two completely distinct watery worlds separated by the plasma membrane. You have the extracellular side on the outside of the cell and the cytoplasmic side on the inside. The massive problem here is that things cannot just easily cross over that oily lipid barrier in the middle. And well, that brings us to our solution. Section 2, anatomy of a transmembrane protein. So the crucial point is, how does this protein actually pull this off? How does it smoothly insert itself through the entire lipid bilayer? Well, it manages this by literally existing in two different chemical worlds at once, divided into three key domains. Step 1 is the hydrophilic extracellular domain, which comfortably hangs out in the watery environment outside the cell. Step 2 is the hydrophilic transmembrane alpha helix, which spans right through the middle. And step 3 is the hydrophilic cytoplasmic domain, extending back into the watery inside of the cell. Actually, I want to pause right here and really hammer this structural detail home. The hydrophobic alpha helix specifically interacts with the hydrophobic lipid tails of the membrane. It's a perfect match, like puzzle pieces. The protein's anatomy is quite literally dictated by the chemical environment of the membrane it sits in. Okay, Section 3, proteins in action. Now that we know what they look like, let's speed things up a bit and see the active, dynamic jobs these molecules perform. Let's look at our first character, the ion channel. Think of this guy as a highly exclusive bouncer at a club. An ion channel allows specific ions, like sodium ions, for example, to move through a gated channel across the membrane. Its primary function is selective transport. It doesn't let just anything through, only what the cell specifically needs at that exact moment. Next up, our second example is the G-protein coupled receptor, or GPCR. This one acts as the ultimate cellular messenger. Notice the sequence of events here. A ligand binds to the receptor on the outside. This physical binding actually triggers a shape change that activates a G-protein entirely inside the cell. The core function here is signal transduction. It's brilliant, really, because it receives a signal from the outside and translates it into an action on the inside, all without the original messenger ever having to cross the border. Let's move to our third example and see how this builds into something even more complex, the sodium-potassium ATPase pump. Unlike a passive gate, this pump actually requires energy to do its job. ATP is hydrolyzed to provide that energy, allowing the pump to force ions against their natural gradient. This makes its core function primary active transport. Now, what is absolutely crucial to remember here is the precise numerical breakdown. If you are prepping for your exams, burn this exact ratio into your mind. 3 sodiums out, 2 potassiums in. This pump transports 3 sodium ions out for every 2 potassium ions it brings in. It is a highly testable, incredibly important biological fact that you are absolutely going to see again. And speaking of highly testable facts, Section 4, CSIR-NET quick facts. Consider these the core identity traits of transmembrane proteins. Do they span the entire membrane? Yes, making them integral membrane proteins. Because they are integrated so deeply into the bilayer, do they require detergents for extraction? Yes. And what are their main jobs? Transport, acting as receptors, facilitating cell signaling, and cell adhesion. If you understand these points, you understand the fundamental blueprint of these proteins. Oh, and one more quick structural distinction to lock in. While most of the transmembrane proteins we talk about use those hydrophobic alpha helices, don't forget about beta barrel proteins. These are a totally different structural form, and they specifically show up in bacterial outer membranes, mitochondria, and chloroplasts. Knowing where to find each type is a classic exam distinction, for sure. I want to circle back to that laboratory fact real quick. Isolating these integral proteins requires harsh detergents. Because they are so deeply embedded in those hydrophobic lipid tails we discussed, you can't just gently wash them out. You essentially have to destroy the lipid bilayer with harsh detergents just to extract them. This makes studying these proteins in a lab setting incredibly tricky. So why are we obsessing over these specific details? Well, because this information is heavily tested across both the cell biology and cell signaling sections of your exams. They're honestly one of the most important topics you can study. The time you are spending right now locking down these concepts is going to pay off tremendously on test day. To wrap things up, I want to leave you with a thought to ponder. If transmembrane proteins are so intimately tied to their membrane that they require harsh detergents just to be isolated and studied in a lab, how much more is there to learn about how they truly behave dynamically in their natural, unbroken cellular home? The cell is just incredibly complex, and there's always more to discover. Thank you so much for joining me for this explainer. Keep up the great studying, and I will see you next time.